other side of the
Hello, and welcome to Certara Talks. I'm Dr. Suzanne Minton, and I'm the Director of Content Strategy at Certara. Today, I'm talking to my colleague, Dr. Joshua Apgar. Joshua, welcome to Certara Talks. Would you like to introduce yourself?
Sure, thank you. I'm Josh Apgar, one of the co-founders at Applied Biomath, recently joined the Certara team, and I'm really excited to be here, so thank you.
Awesome. Great to talk to you, and so glad to have you in the ABM crew as part of the bigger Certara family. So, Josh, I hear that you recently got back from the Molecular Glue Drug Development Summit meeting. Sounds very exciting. Thought we could talk about it a bit. First of all, a lot of our audience probably isn't familiar with what a molecular glue refers to. So can you talk about that, please?
Sure. So molecular glues are part of a larger family of a new pharmacology of drugs that looks to target intracellular proteins through to the proteolytic machinery as a way of degrading intracellular targets. And kind of the tagline has been drugging the undruggable for them. Molecular glues as a subtype of that are a type of molecule that really enhance existing protein-protein interactions within the cell. And kind of the prototypical example of that is thalidomide, which for a long time the mechanism of action was not known and was later discovered to be one of these types of glues. And really what they do is by fitting into that protein-protein interface, they kind of help stabilize that interaction and enhance the sort of the natural degradation machinery. And so these have found all kinds of life in initially in cancer, but increasingly in immunological diseases, in other places where being able to selectively target a protein that's not necessarily a kinase or one of the types of drugs that are easily drugged really enables sort of a whole new class of pharmacology to be opened up.
Very cool. It's exciting emerging technology. As we were talking about earlier, when I was in grad school, I remember learning about E3 ligases, and that basic biology was just starting 20 years ago, and now these molecules are starting to make it into the clinic. Can you talk about what you think are the challenges for molecular glues to get into the clinic and run through the drug development process to become approved therapies for patients?
Yeah, well, as you can imagine, these molecules that have to fit in at these very compact protein-protein interfaces, the chemistry has been really a big challenge there, and getting enough interaction without disrupting the existing interaction has been really a challenge. And if you went to the conference last year, a lot of the focus would have been on that. But as things are starting to move towards the clinic, the other real promise here is that these could be orally bioavailable drugs, that they could be given in a regimen that's very different than the biotherapeutics, which are all injectables. And so that means they have to balance these sort of complex, binding energies with properties like PK, solubility, drug absorption. And so I think that's a place where modeling really, can have a big impact here, is sort of how do you balance these properties? And I think if you looked at a lot of the topics of people talking about the drugs that were sort of at that transition from preclinical to clinical, those are really the challenges that they were wrestling with, which is things that improved the glue effect often had negative impacts on other aspects, and figuring out really how to balance those. And so I think that just, one, reflects the maturity of the field as a lot more molecules move into clinic, which is very exciting, but also that they're having to tackle that next round of challenges.
Absolutely. And the Applied Biomath team, you guys are QSP, quantitative systems pharmacology experts. Can you talk about how ABM is using some of your QSP technology for this specific problem of molecular glues?
Yeah, so some of it's around simulating the primary pharmacology. So really just looking at if you have to trade off, half-life or absorption or solubility for binding potency, what are the optimal places to trade off there so that you get the best possible molecules. The other is starting to think about now in clinical design and dose regimen design, and really understanding, these molecules don't just stick things together, but they target them to be degraded. You have this extended pharmacology of the resynthesis of your target over time, and really using that to understand how best to deploy these drugs and how to sequence them so you have successful clinical trials. So I think it's really bringing all of that together into one coherent picture and allowing teams to kind of make the best decisions given, the various tools that they have to make adjustments.
Absolutely. And it sounds like you're optimizing all these different parameters, like understanding structure activity relationship, but also, having reasonable PK. It seems like these would be very long acting drugs since you have to synthesize the proteins that they're degrading.
Yeah, no, that's exactly right. And so I think these are generally, people are going for oral daily delivery, but the proteins themselves may resynthesize on the timescale of, hours to days. And so I think people are also thinking about things like drug holidays, and do you give a dose every day, or is the reason why twice daily dosing might be preferable, both from an efficacy perspective. But I think like all these drugs that target intracellular targets, selectivity is a big challenge, because the same targets exist in many different cell types, some of which you may not want to target. And so understanding whether there's room to improve therapeutic index through things like the selection of dosing, is another feature where, now we're not talking about simulating the pharmacology, but the sort of downstream biology of the target and what happens after that. And so in that way, it's very similar to the way we employed QSP in other modalities. It's really connecting it all the way back to the properties of the glue, so that we could sort of really tell the sort of start to finish story and understand how the parameters at each level really have impact.
Fantastic. Well, Josh, it was a pleasure talking to you today.
Oh, thank you.
And if you'd like to learn more about what Josh and his team are doing, please visit them at the Applied Biomath website. I'm Suzanne Minton. You've been watching Certara Talks. I'll see you next time.